Okay, this is chapter 16, How Genes Work. In this chapter, you will learn how genetic information flows from DNA, RNA, proteins, and this is called central dogma. So we'll go over what genes do. We'll talk about central dogma in greater detail. We'll talk about mutations, and then we'll focus on the genetic code and how this genetic code is like a three-letter word. So let's start talking about genes. We know that DNA is a blueprint of life, and that DNA is so important that it's only found in the nuclear membrane. It's protected. It's like the inner part of a city, right? It's like the downtown area. That's where all the important buildings are. So we know DNA contains the information that makes us us, and at the cellular level and at the molecular level, it is protected so that it can continue to have that information. But what about a gene? Okay, so I want you to remember that genes are found on chromosomes. So one linear chromosome found in a eukaryotic cell can have many genes located, and genes code for a trait. And usually these traits are defined by proteins. So how did we find out about genes, and how did we go from like having a code, like a computer code, and then you end up with some kind of product at the other end? So how does that work? How is it that you go from code to actually something that you can see? And that in itself is central dogma. So there's a classic experiment done by Beadle and Tatum. Now, I'm not really going to discuss it. I want you to read it in your book. So I'm going to synthesize my understanding of this experiment for you. And if you don't like it, then read it in your book. But what they did is they looked at a metabolic pathway, and they looked at it in bread mold. And they wanted to know like the association with genes on a chromosome with the actual production of this metabolic pathway, like how were they connected to one another? So this idea started forming this idea of one gene, one enzyme. So that means that one gene has a code. It can only code for that one enzyme. It isn't going to code for anything else. That code is specific for that one enzyme. And maybe we can extend that and say it's specific for one protein, which translates into a trait. So they did this experiment, and what they did is they knocked out some of the genes on the chromosome, and they wanted to see the effects on the metabolic pathway. So here's our metabolic pathway, and notice that at each of the steps is catalyzed by a different enzyme, enzyme 1, enzyme 2, and enzyme 3. So when they looked at the results, they came to the conclusion that it was this idea that one gene actually codes for only one enzyme. And we can extend this to one gene, one polypeptide. And remember, guys, what is a polypeptide? Yeah, you're right. It is a protein because proteins have peptide bonds. So how they found this out is by knocking them out or loss of allele function. It's like, for example, when I hurt my thumb and I couldn't use it, then I realized all the things that my thumb could do. I couldn't button my shirt, I couldn't hold a cup, I couldn't write, I couldn't hold anything. I couldn't use my hand to balance myself. So then I learned how my thumb worked. And they used the same thing. Once they knocked out the allele, they understood the loss of function, and that's how they figured out this relationship between genetic information and what we actually see. So using loss of function, we came up, and it supports this idea, one gene, one enzyme, or one gene, one polypeptide. Genes specify production of proteins. So the answer lies in the structure of DNA. So recall that we can find many genes on DNA. So a gene is a stretch of DNA that codes for a protein. So when we talk about this code, what are we talking about, this genetic code? So Francis Crick proposed a sequence of bases in DNA acted as a code, and these codes were actually coding for amino acids. And you say to yourself, hey, Dr. Weissman, weren't we just talking about DNA and proteins? And the answer is yes, we were. But remember, what are the building blocks of proteins? Amino acids. So that's why we're talking about them right now. And amino acids are like letters in our alphabet, right? We can make short words, long words, and then when we string the words together, then we get different sentences that express different ideas. So the same thing goes at the cellular and molecular levels for amino acids and proteins. So we know that there's 20 different amino acids. And each stretch of DNA, or each section called a gene, specifies this amino acid sequence. And this sequence is going to build a unique protein. So we had another group of scientists suggest that RNA links genes in the nucleus to protein synthesis in the cytoplasm. In other words, it's not just DNA protein, it's DNA, RNA, and then protein. So the link between the two of those is what we call messenger RNA. And this is going to lead us to central dogma. So messenger RNA is found in between DNA and proteins in this process of actually going from code to an organism. I really appreciate figure 16.3. At first glance, you might think that this is a very simplistic diagram, but it's a very telling one. It tells of central dogma. It tells the story of how we go from code, how life goes from a simple code with 4 letters, and it produces a protein, and that's what most organisms are made of. So let's take a look. Inside of the nucleus, we have DNA in red, and a section of it will, or some genes will be copied by messenger RNA indicated in yellow. And that messenger RNA will exit the cell, and it's going to find a ribosome. Remember, ribosomes are like little greeny beads out in the cytoplasm. And when it goes out into the cytoplasm, the ribosome is going to start reading, actually the RNA, and as it reads it, it's going to convert it into a protein. And this protein is indicated by the green strand down below. This is called central dogma. DNA, RNA, and protein. I promise you, you're going to have several test questions on central dogma. DNA, RNA, and proteins. So this is how we go to code to actually something that's three-dimensional like us. Okay, so we can break this up into transcription and translation. To go from DNA to messenger RNA is called transcription. Isn't transcription just a fancy word for copy? And isn't that in fact what is happening? Messenger RNA is copying some information from a section of DNA. It's copying the genes for a certain trait. It could be like hair color or coarseness or curliness of hair. So that's what it's copying. And then the second part is translating. It's the process of using information in messenger RNA to actually make or synthesize a protein. It is the interpretation of this that is going to build the protein that is a three-dimensional shape and what makes us, us. So I want you to recall from the previous unit that we did talk already about genotype and phenotype. Isn't it when we talk about genotype, we're talking about genes? And if we talk about phenotype, we're talking about what you physically see. So genotype is like big T, or big T, or two little t's. Phenotype is tall, short, brown eyes, blue eyes. Okay, so here we have DNA, messenger RNA, and proteins. But notice that when we start off with DNA, aren't we in fact starting out with genotype? And when we end up with proteins, aren't we really talking about phenotype? It's what the trait that we see. So remember that alleles are versions of the same trait. Alleles of the same gene differ in their DNA sequence. Does that make sense? Sure it does. Because if you're going to be a different version, then you have to have a different sequence of amino acids to form different proteins. This is a classic diagram, and this is one of my favorites. Like, if you can look at this diagram and really understand it, you understand everything that we've just discussed. So this is about central dogma, DNA, RNA, and protein. It's also about genotype and phenotype. And so it's taking you through transcription and translation. So if you take a look, on one side we see a brown mouse, and on the other side we see a white mouse. This little brown mouse is brown because of the sequence found on a strand of DNA. And notice here we have transcription. It goes from DNA to RNA in yellow, and then from yellow it's going to code for amino acids, which are going to make a protein, which we'll see as the different pelt colors of these mice. So this is what makes a brown mouse brown, and these mice live on the mainland. Now let's look at a mouse found on the beach. Notice that it has a white coat, but how did that happen? If you look back at the very beginning of the DNA strand, there's actually a mutation or a mistake. Instead of G at the very beginning, there's an A. And because of that A on the DNA strand, then the complement on the messenger RNA is now going to be U. And because of that, instead of arginine as the amino acid ARG, we now have cysteine, CYS. So that one point mutation can change the color of a mouse's fur. So notice at the very top, when we're talking about DNA, we're talking about genotype. And then when we get down to the amino acids that make a protein, we're talking about phenotype. This is an excellent figure. Please put a double star for studying. Now, there are modifications in central dogma. It is not always DNA, RNA, protein. In some cases, it's RNA first, then DNA, then RNA and protein. But we'll save this for an upper division biology class and a class maybe that focuses on the replication of viruses. So, but for the most part, central dogma does hold true. DNA, RNA, protein. Now let's focus in on the genetic code. The genetic code is 3 nucleotides together. It's 3 bases. So it's a sequence of nucleotides. There's 3. And it says here that each of these 3 bases is considered like a word. And keep in mind we have 20 amino acids, so they're going to code for these. So we have a triplet code. And notice here it says 4 times 4 times 4 is equal to 64. So where did we get the 4 from? The 4 is from adenine, thymine, cytosine, and guanine. Or if we're on messenger RNA where the code is found, it's AUCG, apple umbrella, chewing gum. So we have a triplet code, and this triplet code covers all the possible combinations so that we can at least code for 20 amino acids. So a codon is a triplet code. Another name for a codon is a reading frame. I always talk about a mutated bunny fufu. Why is it a mutated bunny fufu? Because instead of 2 ears, there's 3. So this is a ribosome reading the messenger RNA. 3, 3, 3, 3, 3, 3. So that is the codon, because the codon is a triplet code. So we can use reading frame and triplet codon interchangeably. Now it says here a reading frame cannot be destroyed by adding or subtracting multiples of 3 bases. So you can imagine this is the experiment that definitively concluded that the codon was a 3-base code. Okay, so we know here's this 3-base code, and they actually did an experiment and they used a lot of U's. So UUU actually codes for phenylalanine, an amino acid. And so this is what they got when they used a triplet code of U's. Now, the one thing that you are going to have to know for the test is out of those 64 combinations, one of those is a start codon. And this start codon is AUG, and it codes for methionine. And it's always at the very beginning. And so you will need to know this for the exam. But do you know what my little mnemonic device is when did the semester start? Yeah, that's right. It started in August, AUG. Okay, so you don't have to know the stop codons, but you do have to know that there's 3 of them. So if I see you doing something and I want you to stop, I just simply say, hey, stop. Stop doing that. But what do you say at the molecular level? You have to use the code UGA, UAA, or UAG to say stop. Stop making a copy. The other 60 codons are used for coding for amino acids. We are going to do an exercise in class so that you familiarize yourself with this table, because as you can imagine, you're going to have to use this table for your final exam. So notice here on one side you have your first base in red, and on the top in blue you have your second base, and on the other side you have the third base in green. So on this table you can find out what the triplet code is. This is the code, and it's been broken down so that you know. Now I've had some students ask me, come on, Dr. Weissman, how come is it that leucine found in the first column near the top has 6 different ways of actually coding for leucine? UUA, UUG, CUU, CUC, CUA, and CUG. And then you have others like glutamine, and they only have CAA and CAG. What's up with that? And to be honest, I don't know the answer. But we can see that the code is redundant. So some have like 6 different variations, some only have 2, but the truth is each code only specifies that particular amino acid. If we look at the code, we can say that the code is redundant. That means all but 2 amino acids are encoded by more than 1 codon. It is unambiguous. One codon always codes for the same amino acid. It will never code for another one. It is non-overlapping. Codons are read 3 at a time. There's a reading frame. It is nearly universal. In other words, if we take some of my code and put it into a bacteria, the bacteria should be able to read my code and put string amino acids together to make proteins. And lastly, it's conservative. Conservative to me is almost like it's redundant. If several codons specify the same amino acid, the first 2 bases are usually identical. So, in other words, most of the time, the third base isn't as critical as the first 2. The first 2 is important, and this last one is not as critical. Now, there's this video is actually in one posted already in Blackboard. So please go ahead and look at it. I myself get confused because notice it's AAT, AAT, AAT. So it's a little bit confusing, but if you like it, you go and see if you can learn something from it. Okay, what is the value of knowing the code? Well, if you know the code and you understand central dogma, then you know how you can go from code to an organism. And to be honest, we already have some synthetic organisms. There's been people that have already built unique bacteria because they understand the code. and they can manipulate the code to build their own organism. So the question is, should that be done? And I'm glad that you're taking this class so that when you vote on things like this, then you will be an informed voter. So if we can predict the amino acid sequence, then we understand this relationship, this intimate relationship between DNA and protein. Okay, so let's take a look at this. Let's do, the template strand is going to be the bottom strand. So we're going to look at the bottom red strand so that we can build a messenger RNA. And notice, messenger RNA is built in the 5' to 3' direction, and that should be of no surprise to you. So the first thing that we have to do is we have to transcribe. So TAC becomes, you're right, AUG. GAC becomes CUG. So let's see if you can finish the rest. And let's see if you can go back to the table and you can put in the appropriate amino acids. So what are the consequences when there's a mistake in transcription? So you can have a mutation. Now there's two types of mutation. You can have a point mutation, which is the result of one or small number of bases, or you can have a chromosome level mutation. So when we looked at the mice and we saw that there was one change in a base, that was a point mutation. But we can also have a chromosome level mutation, which would be like Down syndrome, and that is caused by non-disjunction. So let's take a look at a point mutation. So in figure 16.7, notice here in gray, we see the original DNA strand, and we see that there's complementary base pairing. We see that the strands have been unzipped and a new strand is being made, but notice here the complement to G is C, but by mistake a T is placed there. Then we have a permanent change, and this is called a mutation. This is called a point mutation. Now there's different types of point mutations, and you will need to know this for the final exam. There's missense, silence, frameshift, and nonsense mutation, and they all mean something else. So a missense means you change an amino acid in a protein. A silent mutation means there has been a change in base, but it has no impact on the amino acid that's building the protein. Then you have a frameshift mutation, and this is due to an addition or a deletion. And then a nonsense means that the point mutation specifies for an early stop, and the protein or the amino acid sequence is so short it has no meaning. It's nonsensical. Now, point mutations can be beneficial, they can be neutral, or they can be deleterious. Remember, we watched a video in lab, and Sean Carroll actually mentioned that we're born with 40 mutations, but now you realize that most of these are silent mutations, that change in the base actually had no impact on the amino acid sequence that was building the protein. Now, beneficial mutations increase fitness, neutrals have no effect, and a deleterious mutation decreases fitness. Most of the times we think of mutations being deleterious, of lowering fitness, but in some cases they can be neutral or beneficial. I love table 16.1 because it summarizes in a very nice manner all the different types of point mutations. And as I mentioned before, you'll need to know this for your final exam. Now let's talk about the other type of mutation, the chromosome level mutation. So there's different types. So I'm going to start off with a story. I had a student a long time ago, and she was very religious, and that's really important to the story because she knew in her family that they had a terrible genetic disorder. And what had happened is one chromosome had broken off, and reattached itself to a neighboring chromosome. So about 50% of the time, as the gametes, remember the law of segregation, separate, she had a 50% chance of having a child that had um, no genetic disorders. But she also had a 50% chance of having a child that would have dramatic um, disorders, and that would make the baby, physically and mentally challenged. And so even though she was religious, she made sure that she went to a genetic counselor so that she could have healthy children. So what happened is now that I've told this story, you know that she had an inversion. A segment of chromosome broke off and it flipped around. And in her case, it didn't rejoin the same chromosome, it rejoined with another one. So here you can have translocation. It breaks off and attaches. So she had an inversion and a translocation. Then you can also have a deletion. Remember, we have non-disjunction. We can have sticky chromosomes, or we can lose just segments of a chromosome. And then lastly, we have duplication. A segment of a chromosome is present in multiple copies. So here they give you examples of all different types: deletion, inversions, duplication, and translocation. But all of these are very serious genetic errors because they're affecting the whole chromosome, not just a point mutation. Chromosome mutations are covered and studied here because we want you to understand that they can be beneficial or have a negative or neutral impact on our DNA. Oftentimes, cancer cells exhibit deleterious chromosome mutations. And so all of these aneuploidy, inversions, translocations, deletions, and duplications can cause cancer. And we can figure this out oftentimes by doing a karyotype or mapping our chromosomes. And here you see that there's a lot going on here. And this particular cell has a lot of mutations. So you can tell this by the color, the numbers, and the length of chromosomes. And this is the end of this chapter. Thank you for listening.